science. He's at the other side of campus, and we don't get to see those folks often enough. Carl's an assistant professor, and he got his PhD at Massey University in New Zealand, and then we were just talking about his postdoc experience at University of Illinois. And he looks at microbial ecology within animal systems, so with a different look at the environment. So thanks for coming.
Thank you for the introduction. And as Anne mentioned, I'm an assistant professor, and our group is specifically interested in microbial ecosystems that live in and on host species. And we study it across a number of different animal systems, all the way from insects, where we're interested in a microbe that lives within the wheat stem sawfly, which is a piece of the wheat industry, through fish, where Lola, one of my graduate students here, we're looking at the microbiota in the gut of rainbow trout in the context of agriculture and climate change, to domestic livestock species, where we're specifically interested in microbial relationships to productivity and health, including immunological health, where microbes are very important in maturing host immune systems, and all the way up to humans, where we're specifically interested in reproductive tract microbes that have a very important relationship with a number of pretty significant urogenital conditions. And while over the past 4 years I've preferred to talk about that research, because that's really the research that we're leading and pushing out of the lab, one project that I've been involved in a little bit more peripherally over the past 4 years since being here at MSU, but beginning as a postdoc at the University of Illinois in Urbana-Champaign, is looking at primates, and specifically our objective was to integrate microbiological understanding into human or hominin evolution. And the project funded by NSF was built around, at least initially, this expensive tissue hypothesis that was elaborated in 1995 by Leslie Gillio and Peter Wheeler to explain these dramatic increases in brain size. I apologize that that didn't work. That we've seen through human primate evolution. And so this is a characterizing feature of human evolution, but it's also somewhat of a puzzle. The brain is, as many probably realize, one of, if not the most expensive tissue that we have, and so theoretically there are constraints in our ability to evolve a larger brain. And so the hypothesis presented by Leslie Gillio and Peter Wheeler was that to enable or facilitate the development or stabilization, which is essentially size of brain normalized to body mass, which is widely considered or accepted as the measure of intelligence, may have been facilitated by reductions in other expensive tissues, and there are specific hypotheses built around the gut, which is, like the brain, a very expensive tissue, both costing around about 20% of our daily energy requirements. And so in the original paper, they were able to show this. It's been studied both in primates, but also in a range of other animal species since then. And while it tends to hold true in primates, and the paper published as recently as 2006, they showed that this relationship was valid, whereby primates that we diverged from further back in our evolutionary history have much larger guts, much reduced brain size, and that has increased over our evolutionary history into humans, where we have a very large brain size and a far reduced gut size. But it hasn't held true in a range of other animal species, including birds, as you can see here. And additionally, in a recent study that came out a few years after we actually began this project, where they looked at 100 different mammal species, including a variety of primates. They actually found greater relationships between brain and adipose deposition. So regardless of the future relevance of that hypothesis, it's interesting to consider the fact that over our evolutionary history, brain size has expanded and has undoubtedly come at a great cost energetically. That the main organ through which we obtain energy, our gut, has decreased quite substantially and is around about half the size of what we would expect if you were to compare it to other primates in the lineage. And while our evolutionary history has involved various bits, there was evidence for various things that have occurred in our evolutionary history to support greater energy extraction from our diets, such as increasing carnivory and production, utilization of lower quality, high and fibrous plant-based feeds, and the utilization, harnessing the utilization of fire to cook those feeds, which both is often shown to not only increase the accessibility of nutrients within the feed, but also make them more valuable nutritionally. One thing that potentially has played an important role is our microbial, who had very fundamental roles in our nutrition, as well as for other mammal. Okay, and so to show you explicitly one of the more important roles that the microbes facilitate in providing us with nutritional energy. This is a metabolic map that you can find on a couple of different websites, and this particular one is from the KEG website. And basically what this is showing is these nodes or dots represent different metabolites, and they could be sugars or amino acids or just about anything that occurs within our metabolic processing. And these edges or lines connecting them represent enzymatic pathways. And so we don't possess all of these different pathways. And if we look at humans, they have some subset, which we can assume by genome. And one surprising thing you might find is that if we compare humans to related animals such as cows, the biochemical processes that they can endogenously facilitate are not much different. And of specific interest is in carbohydrate breakdown, right? So if we zoom in here, now we're looking at the same pathways, but here our enzymatic capabilities are shown by these highlighted enzymes, which are enumerated using in ontology. We can see that when it comes to the breakdown of starch, for example, we can do that very efficiently through glucose, a simple sugar that we can utilize very efficiently because we have the enzymatic pathways. Whereas if we look at some of these more complex carbohydrates, cellulose, for example, you can see that none of those enzymatic pathways are encoded within our own genomes, which means we don't have the ability to break these things down from where they arrive. Likewise, xylin, another major polymer within plant-based foods, is indigestible. Pectin, other things that are grouped in with xylan, which is a predominant, considered a hemicellulose. Mannans are equally indigestible, and fructans as well. And so if you consider fruits or vegetables or any plant-based matter, these polymers basically make up the vast majority in these nutritional resources. And so if you take those out of the picture and also ignore lignin because it's equally indigestible, it doesn't leave a whole lot that you can extract from these food sources. By contrast, the collective assortment of microbes that live in our guts possess all of the enzymatic functions necessary to break these things down. And they break them down into simple sugars, which they then ferment into these things called volatile fatty acids. The three most abundant being acetate, propionate, and butyrate. And these things are basically currencies of energy that the microbes provide to the animal, the host, and can be utilized as a source of energy. They also have a large number of other physiologically important contributions as well to health and other things. And so if we consider, for example, a ruminant animal, such as a cow or a sheep or an elk or a bison, because of their strictly herbivorous diets, they actually depend on microbially produced short-chain fatty acids or volatile fatty acids for around about 70% of their daily energy requirements, which means that they basically couldn't exist without their microbes. By contrast, we consume a highly refined, much more high-quality, simplistic diet. And so the microbes contribute a whole lot less, but they still contribute somewhere between 9 and 11%. And then if you look across the primate spectrum, they fall somewhere in the middle. So gorillas, for example, around about 60%, and howler monkeys somewhere around about 30%. This is just to say that in addition to providing that energy, microbes also contribute nutritionally. In other ways, they produce 9 of the 14 different things that we recognize as vitamins, including all of the B and K vitamins. And so if we think about microbes in our own evolutionary history, it's important to recognize that they were the first organisms on the planet. So if we consider the time from the origin of the Earth some 4.5 billion years ago through to today, microbes not only existed in the fossil record less than 1 billion years, or a little over 1 billion years after the origin of the Earth, but they existed in structurally organized communities that were sufficient to be identified using these methods. By contrast, they existed some 3 billion years before animals did. And one of the earliest animals in the fossil record is a mollusk-like organism known as Kimbrella, which occurs only around about 500 million years ago. And then if we consider early humans or hominids around about 2 billion years ago, 2 million years ago, and then anatomically modern humans around about 200,000 years ago. So irrespective of where you consider our story beginning, we've had to evolve alongside microbes. And there are numerous, so we've had to evolve in a sea or a swarm of microbes, right? So if we just consider bacteria, which is just one type of microbe, not only do they outnumber all of the people on the planet, all of the animals on the planet, but they also outnumber all of the stars in the observable universe. In fact, there are so many that, to put it in perspective, if you took every one and lined them up end to end, recognizing that microbes by definition are entities that can't be seen with the naked eye, they would span more than 2 billion light years. That's how many microbes we're swimming in. And consistently, they're also the most abundant cell type in and on our body, and contribute far more to our genetic capabilities than we do ourselves. They outnumber our own cells by an order of magnitude, and they contribute somewhere between 100 and 150 times the number of genes that we endogenously encode. And obviously, as I've tried to point out already, these genes are contributing to our way of life. So this project began sort of early on in the microbiome era, where technological development was enabling interrogation of these host-associated microbial ecosystems on an order of magnitude that was previously unprecedented. Right? And so one of the earliest studies that came out in that era came from Jeff Gordon's lab from a researcher named Louis Play, where she looked at a number of individual animals that were kept in a couple of different zoos in the US. And what she found, and what is important to keep in mind, is that these microbes, or these microbial ecosystems, showed relationships with diet and host phylogeny. So one of the first observations that we made in our collection of studies was that primates had these species-specific gut microbial ecosystems that were equally divergent between very closely related species. And so if you consider here two species of colobus and another old world monkey, you can see that in this non-metric multi-dimensional scaling plot, which basically, you actually interpret this, these individual figures represent whole microbial ecosystems, and their distance from one another represents how similar or dissimilar they are based on diversity, which is presence and abundance weighted. You can see that each of these form very distinct groups and distinct from humans as well. So much so that in a study that we haven't yet published, looking at baboons that were cohabitating in a region of Western Africa, the two species of baboons, Hamadryas and Nubis, form very distinct clusters here in blue and green, and occasionally these baboons actually mate, interbreed, and form these hybrid species that look something like this. And you know, it's quite fascinating that these all right in between, suggesting that there's some sort of genetic component structuring that microbial ecosystem. And perhaps you can see it a little bit better here. Now we have another baboon, or baboon relative as an outgroup, and you can see that these two populations are distinct, and these hybrids tend to fall right in the middle. And so as we've gone on and added more species to this understanding, you can see that this host specificity tends to hold. We're humans, and regardless of their origins, these are white humans that have moved to Africa. These are two different populations in Africa, all cohabitating, and you can see that they form microbial ecosystems that are more similar to each other than other primates. We're then more similar to apes, who are our closest primate relatives. And then, you know, more similar to the old world primates, which go down our own lineage, then we are to new world primates, and prosimians are even further afield. But one thing that we did notice in putting this together, beyond the fact that these primates are geographically, you know, spaced or occupy different regions typically, is that if you compare captives and wild populations, you see very striking differences. And in this particular figure, you can see here with vervets, we have two populations here, one captive, one wild and free ranging. You can see they're significantly different from one another. And in howler monkeys, zoo kept, some zoo-like conditions, and wild, and you can see that putting them in captivity makes them much more like humans.
But the races there in black indicate that different races, quotes, do have different compositions.
Yeah, there are certain patterns, and I'll certainly talk about that later. But if you're comparing, and you know, certainly if we're talking about individuals, even us within this group, you know, your microbiome is going to be very different from your microbiome, but both of your microbiomes are going to be more similar to each other than a different species of animal, and that's really.
Can I ask you a quick question, Carl?
Yep.
The ones that are the monkeys that you're comparing that are captive bred versus non-captive wild monkeys. Do you know whether those are fed captively on formula versus mother's milk? There's a lot of lipids
You're
that
talking
have been
about
associated with
early
microbiome
which
establishment,
is
and I'm
something
just
we're very
wondering.
interested in.
Yeah.
I don't specifically know these particular populations, but there are certainly dietary differences, and it's something
Right.
I'll talk a little bit more about.
Okay, just curious if you
Yeah.
knew with those guys. Yeah.
That's a good point. Okay, so sticking to the theme of diet, primates represent a very interesting comparative lineage because of the dietary variation that occurs within them, right? So ignoring us who are somewhat different from everything else, primates might prefer fruit, they might prefer leaves, they might prefer insects or small rodents or nuts or other foliage. And those dietary variations don't always match biology, right? And the other important consideration that we showed in the paper 2 years ago now is that the microbiome's response to diet appears species specific as well. So if we consider humans who are consuming, these high fat, high sugar, high salt-rich diets that we often describe in the literature as Western style diets, it is well known that this invokes a pretty dramatic shift in the gut microbiota, lowers diversity, shows a number of features that we would describe as dysbiotic or that correlate with various diseases or disorders. But if you take vervet monkeys and compare one population to a second population that is consuming a typical Western diet, you might think that a diet was, the primary shaper of the microbiome, that might become more human-like. But in actual fact, they shift in a very different direction, and by some metrics, they actually appear less similar to humans when consuming their diet. Okay, so therefore it's important to explore environmental gradations within species, within a species, and we wanted to do that within specific locations as well, or close locations. And so the first study, which finally got published in 2013 after bouncing around a couple of journals, undertaken by Catherine Amato, led by Catherine Amato, who was a graduate student in the lab when I was there. She's now has a faculty position at Northwestern. And she was following 6 groups of black howler monkey around Blanquet National Park. 2 of these groups occupied a continuous evergreen rainforest. And this is considered most consistent with the archetypical niche or the environment that they'd most likely have spent the majority of the evolutionary history in. 2 groups that were in a semi-deciduous forest, resembling this, but an encroachment of trees that lose their foliage for part of the year. And then another group that was found within a fragment, and so as populations move in and deforestation occurs, it creates these almost like islands of trees. And because these particular species are arboreal and try to avoid coming down to the ground due to predation, it basically locks them up in these environments. And then captive populations, which actually originate out of most of this, but also these populations as well. And so we asked what are the relationships of these microbial ecosystems. And the first thing that became evident is that as you go down these different gradations in habitat quality, you can see that primates, or the black howler monkeys that were occupying this archetypical like niche, had far more diversity than anything else. And then everything else seemed to fall down from the semi-deciduous to fragment, and then captives had the lowest amount of diversity. And again, diversity is one of these things that we can associate, or we often associate with disease phenotypes.
The diversity is all in the gut. You're not looking at anything else.
This in these studies, yeah. Not in this study, but in a number of the studies, we looked at the reproductive tract as well. But I'm not going to talk about those today, but certainly can any other time. Okay, but comparing them in terms of their composition, this was the first plot we put together, and what might not be obvious in this is while this is all of the captives sitting on top of each other, this is actually every other primate from every other habitat that is forced onto each other because of the striking difference that exists between these two groups. And it's significant using analysis of similarities, which is a measure of how dissimilar these two groups are. To then ask if these other habitats were equally divergent, it was important that we remove these captive individuals. And that's what's down here. And so you can see, looking here, the continuous evergreen rainforest communities were significantly different from either the fragment or the semi-deciduous rainforest groups. And those two groups were different from each other as well. Okay, so one of the great things about this study, Katie spent a lot of time down there and was able to follow 4 of these groups over that time and observe their dietary habits and patterns, and then quantify them as well. And that was something that she has been training to do as a graduate student. And so what she was able to show was that, pressing on the buttons and getting ahead of myself, but what she was able to show was that the monkeys occupying this continuous evergreen rainforest were consuming very different species of plant and very different parts of those plants than those within the semi-deciduous forest habitats. And if you want to look at that quantitatively, those within the continuous evergreen rainforest were consuming, on average, from 15 different tree species versus 9 different tree species in the semi-deciduous forest. The diets were primarily mature fruits versus foliage, the more folivorous diet of the semi-deciduous animals. Unfortunately, she wasn't able to collect quantitative data from the other 2 groups, but a qualitative assessment of their habitats suggested that the fragment had a very similar set of plant species, although it was reduced in number, and captives were actually fed a commercial monkey chow, some fruit, and some yogurt. Okay, and so in looking at just the semi-deciduous and continuous evergreen rainforest groups, Katie was able to show using Mantel tests that there were significant relationships between the diets that they were consuming in terms of both the plant species and the parts of the plant that they were consuming and their microbial profiles, suggesting that diet was probably the biggest factor driving these differences. What was also interesting in this observation is that if you look at the species of plants that they're consuming, you can see that these individuals have very dispersed patterns of what plants they're consuming more regularly, what parts of the plants they're consuming relative to one another within the continuous evergreen environment, relative to those within the semi-deciduous environment. So you might expect that consuming those different plant species might increase the variation within their gut microbiota. But in fact, the opposite was true, right? So the monkeys that were in the continuous evergreen rainforest were consuming more different, highly varied species of tree and plant types that had much more similar microbiota to one another than we were observing in other populations. And if you look at that, we used Ray Curtis in this particular study to compare microbial ecosystems. You can see that the relationship of the microbiota in this population far greater, or is greater than in the other populations. Okay, so that leads me to a second study that was in a species of New World monkey. We wanted to get closer to humans, and so another graduate student in the lab at the time, Andreas Gomez, who's now at the J. Craig Venter Institute, was afforded the opportunity to travel to Africa and, along with a collaborator, follow 4 different groups of gorilla that were approximately close to one another, within sort of 5, 10 or so kilometers. But what was interesting about these 4 groups is that 2 of them were in various stages of habituation. And so what that means is habituation is basically the process that is undertaken before ecotourism can take place. So it's exposing primates to humans, making them comfortable with anthropogenic activity. And so one of these groups, H1, which you can see down here, was actually on a daily basis exposed to human traffic. It was completely habituated. Whereas H2 was just starting to be habituated to deal with the increasing ecotourism that was going on within the park. Okay, so, and the other point is, there were 2 groups that were completely unhabituated. And so you might ask how they were able to collect samples from populations that weren't or really exposed to these animals. They actually knew where their nest sites were, and they would wait, and then within the early hours of dawn after they left the cave, they would go in and scoop up the feces, which there was a silverback in each of these tribes, which is the big guys, and something that I wouldn't necessarily want to be doing, but they did it anyway. And so for the habituated groups, Andreas was able to replicate Katie's methods and explore what their dietary patterns were. And it's depicted here, H1, again, the highly habituated group that was occupying this more southern region of the park, were predominantly or had significantly greater intake of leaves and insects relative to H2 up here, the group that's just becoming habituated, who were consuming more food. And this was really driven by resources available within those 2 regions of the park. Yeah. Okay. And so, as you might expect, well, maybe not expect, but variation within their gut microbial ecosystems here using a canonical correspondence analysis showed that the 4 groups separated out. That was the strongest factor driving apart these different microbial ecosystems. But there was also a clear delineation between animals that were occupying the more central part of the park and those occupying the southern part of the park. And perhaps what was more interesting in the study was that the habituated group, the one that was exposed to the most anthropological pressure, was the group that really separated out from everyone else. And you can see that more clearly down here, but perhaps also up here. Suggesting that, that anthropological pressure was contributing in some way, perhaps through stress or something else, to driving those microbial ecosystems apart. Andreas wanted to know what that meant functionally, and so he had a look at the metabolomes and separately the short-chain fatty acid profile, remembering that the short-chain fatty acids are basically the currency of energy that the microbes provide to the individual. And what he saw was that unlike the microbiome, which was separating mostly between groups, but especially with the anthropogenic activity, short-chain fatty acids were, well, first of all, the metabolites were varied by location, which you can see here. But the short-chain fatty acids, basically reflecting the diet. So those that were consuming that foliage-rich diet, which would have been higher in those complex carbohydrates, had much or significantly elevated levels of short-chain fatty acids, particularly in acetate and propionate, which tend to contribute a little bit more to energy. Whereas butyrate, at least this is a relative proportion, was higher within those that were occupying this more central region and consuming those fruit products. And butyrate actually is probably one of the most important metabolites, contributing in various ways to our health.
Now, that does not imply that the plants contained those. That implies that whatever they contained
The
was
plants
converted
definitely do
into
not
the
contain
acetates
this.
and the butyrates.
Yep, this is a microbial product that's derived in here. Other metabolomic differences we're seeing, and these include things that you might expect from the diet, increased sugars in the fruits, and other things. Okay, so those primates, old world and apes, a little bit close to us. But what about humans? Andreas had an opportunity while he was over there to also sample cohabitating or closely habitating human populations that represented two different levels of resource utilization. I'm trying to think of their word. But hunter-gatherer population, Baka, Pygmy tribe, and the Bantu, who were very closely situated population of native African people that had adapted to agriculture. And so it presented a very interesting comparative opportunity, again, to look at lifestyle and habitat. And so, interestingly, what he saw, this is using a slightly different analytical approach to compare microbial ecosystems. Basically, this is using a metric known as phylodiversity or phylogenetic diversity, whereby not only are you looking at the relationship and the composition of the two microbial profiles, but you're taking into account the relationship of the individual bacteria within those populations as well. And using an unweighted measure, whereby basically you're asking in a presence or absence of specific microbe context, he didn't see any difference between these two populations. But if he made it weighted, which means that now we're looking at the relative abundances of these specific microbes within these gut populations, you can see that there are differences, and in fact, that is significant. And so you can see those even way back at the phylum level, most explicitly through the differences in the relative abundances of two phyla that tend to be the most abundant within gut systems, Firmicutes and Bacteroidetes, whereby the hunter-gatherer population had typically higher levels of Bacteroidetes, relative abundances, relative to the Bantu, where it was about 5-1 Firmicutes to Bacteroidetes. And so this is an interesting observation because, way back 10 years ago, I guess that's not way back, but some of these early studies looking at gut microbial ecosystems found relationships between these two phylum-level populations and obesity, and whereby increases in Firmicutes related to obesity and similarly decreases in the relative abundances of Bacteroidetes. And so these studies have held true in many analyses, but there are also a handful of studies where they don't hold true as well. And so it may also reflect differences in nutritional status. For example, we know that in hibernating animals, including bears, that when they're hibernating, we see decreases in Firmicutes, or relative abundances of, and increases in the relative abundances of the Bacteroidetes. And similarly, if you look in a python snake, which feasts very occasionally, you can see that the relative abundances of these two phyla change with feeding, whereby Firmicutes becomes far more abundant after a feeding meal, a feeding event, and is far less abundant, towards the end of a starvation period. So, if we put that back into perspective, if we consider that increases in Firmicutes represent nutritional status, then the bucca, which we know can go periods without having adequate nutrition. They're a hunter-gatherer population. It depends on their ability to harvest game and collect other resources. This could be what you're seeing here. Okay, so I guess the last comparison then that I want to make to these populations is to modern westernized, I want to say modern, I didn't want to say modern, to westernized communities. And so in this particular study, we looked at American individuals in comparison to the hunter-gathering and early agricultural populations that Andreas was able to survey in Central Africa. And what you can see is that there are very striking differences between US Americans and these other populations, using either unweighted or a weighted metric. And this actually illustrates those relationships whereby you can see the greatest differences between these two populations of Americans, and then between the two populations themselves, and then within population comparisons using either metric.
What are the coordinates there?
These are uniprot-based analyses. So these are basically it's a PCA button, so it's a measure of distance between these communities. Okay, so then putting that on a canonical correspondence analysis to get a sense of which particular taxa were being affected through these three different populations. You can see that Bacteroides, which is a well-described, probably the most predominant member of our gut systems, is traded out typically for Prevotella. They both are from the Bacteroides family. But there are also this large diversity of changes that, or large number of microbial taxa that are more common or more abundant within these populations than within American populations. And consistently, if you compare diversity of the number of different phyla within these samples, you can see that Americans have far fewer different types of organisms that you see in these other populations. So basically, that's just I guess a series of vignettes that kind of stitched together nicely around that project. And so, in conclusion, gut microbiota are species-specific, host species-specific. They play important roles in nutrition as well as other things. There are changes in the gut microbiota that correspond to habitat variation, and those changes are seen both in the composition as well as the diversity or the numbers and relative abundances of those species, perhaps in the functional ability and the stability as well. But these changes appear to be related to diet, and those diets are often driven by nutritional resources available. But it's important to recognize that the responses are not always identical when you're comparing different species. And so one question that I think we really need to start getting at is, do these changes that we observe represent adaptations to the diet, some form of plasticity of the microbiome, or is this just a reflection of some dysbiosis where all of a sudden the microbiome can become more stochastically colonized? If so, then given our short evolutionary history from the scale of things, and particularly our dramatic recent changes in dietary regimens, it's interesting to ask if the low diversity and highly variable microbiomes that we see in humans is a consequence of a constant state of perturbation that we're in while we're adapting to our new environment and diet. But yeah, with that, I thank you for your attention and happy to answer any questions.
If you go back to the preceding slide. So if you started introducing the red microbes into these individuals here, or the blue ones, if you like, what would be the success of doing that? You could introduce them with or without a corresponding change in diet.
Right. Yeah, and certainly, there's studies that are more easily performed within like a germ-free animal type condition. And studies that people are interested in, although it tends to be within specific microbes rather than whole populations.
Just a quick question as a mother, and then also as a researcher interested in antimicrobial peptides as novel ways of approaching bacterial biofilms. I've really gotten into a lot of the importance of nursing and what components are in mother's milk. And one of the things that's known between the Baka and the Bantu is the difference between how long they nurse. So the Baka nurse for about 4 years versus the Bantu, which nurse for a short period. And of course, we're even shorter in the USA. Have you guys looked at any of those components? And again, I keep coming back to the lipids, but the lipids that are metabolized by bacteria and certainly not metabolized by us, and yet we produce them in sort of our nutritional source for our babies.
We haven't looked at those things. I mean, the lipids are certainly one thing, but the human milk oligosaccharides or the oligosaccharides
Right.
are also pretty significant.
It'd be interesting to see how much that accounts for what differences you're seeing in populations.
So, the importance of that point is that, the human gut microbiome matures over a period of around about 3 or 4 years, right? Breast milk it both provides immunological stimulants and protective features, but it also has microbiome shaping carbohydrates and potentially lipids as well. So, so the period of time that occurs over could potentially be very important. But I'm not familiar with.
Do you know if that's something that he captured in his data? As far as when doing the metabolomics, did he profile the non-polars at all, or do you know?
Yeah, I'm sure it's there.
Yeah. It's data you could go back to potentially.
Yeah, I'm just not sure if we're stuck out as important. And these, I should also point out, are in adult individuals as well. So
Okay.
that is 28 and 29 each.
What would be the effect of seasonality? You see so deer, cattle, we might have big differences in diet among seasons.
Right.
And
In a follow-up study to the howler monkey study, Katie actually followed the populations of bacteria once well, she was there for a year. There was a paper that we published last year before, basically looking at that. And so what she did see was that with the dietary changes, there were corresponding changes in the microbiome as well. So I guess trying to answer your question for sure, over time, depending on resources, things are going to have different influences on the microbial population.
Does the microbiome, is there a change that compensates for the reduction in the gut size? Is there any way you can
You're
look
trying to
or
go back to
think
the
about that?
It's a difficult question to answer. In the most recent paper, one that I was talking about previously, Katie tried to suggest that microbes might be differentially contributing to energy based on, or trying to think of the right wording, but contributing to differences in energy demands in different ways. I'm not sure that evidence for that is there. I think it's kind of hard to ask that question. I think there's some major limitations in that you already have extant species to look at, and they don't necessarily fall quite in line with your current evolutionary trajectory that you just got divergence from.
It's interesting to think about it.
I mean, right from the
Like,
start,
even if you
I
can't
realized this
answer
project was pretty hard to. Honestly, but you know, it provides some interesting opportunity for comparative analysis.
Not to be a question hog.
You are though, let's see if there's anyone else.
Maybe we were trying to pull apart causal inference or a causal association between the microbiome and obesity, as opposed to it just being, I mean, what comes first? Does the obesity change the microbiome,
Oh, yeah.
or does the microbiome change weight?
Yeah, I mean, that's a criticism in a lot of, especially the early microbiome work. There's very good evidence for role of microbes at least in certain forms of obesity. And you know, some of the best evidence for that is that you can take microbiota from an obese mouse and put it in another mouse and make it obese. Versus from a lean mouse, doesn't have the same effect. You can do it from a human to a mouse and get similar effects. If you feed a germ-free mouse a high-fat diet, it doesn't get obese. Whereas if it has a microbiome, it does. And some of the most interesting, I think, or compelling mechanistic data comes out of a group in Belgium where they see similar relationships if they, instead of providing a microbiome, they provide LPS, and they see similar adipose tissue or inflammation in adipose tissue that leads to phenotypes almost identical to obesity. So there are whole theories around proteobacteria, which has an LPS that appears far more pro-inflammatory than other.
So you have to be careful what probiotics you take.
Yeah, well, typically they're not part of a probiotic. So, I mean, there is good evidence. Some people would argue that there still isn't any conclusive evidence, but I think in terms of the different things that people suggest the microbiome is important for, there's probably more evidence there than most, in my opinion.
What's the underlying sampling? You just need fresh feces.
Yeah. Yeah, so you have to be careful, right? You don't want to pick up environmental microbes. Microbes are everywhere, and so in these particular cases, most animals were collected from fresh feces, and there was much effort. They were collected off the ground, for example, to try and collect them from within the fecal matter. One of the most interesting collection approaches was in the black howler monkeys. So they're arboreal, and they tend to spend almost all of their time in the trees. And so Katie actually had a net and was catching them doing her observation studies. And then there are others that we just couldn't get at, and we had to collect, or we did collect urogenital tract samples as well. And so in those instances, the animals had to be sedated for collection purposes. Yeah, so there are a variety of approaches to getting those samples. And then, you know, as I mentioned with Andreas's work in the and gorilla species, the unhabituated ones, it was sneaking into the cave and then 3 hours of dawn, 3 hours of them leaving the cave and grabbing a sample as quickly as they could.
Well, Carl, thanks so much. This has been really fascinating.